So first of all, I would like to thank the ToxMaster Class initiative and the people who are managing it, including Alex Thomas, Hanumant, Yu Sanju, and all others whom I also don't know. So this is a great initiative, and I regularly see the efforts that people put in inviting people, then making WhatsApp groups, and then organizing these lectures. So thanks to all of you who are doing putting all these efforts to organize this such a great program, and especially to the speakers also who will be joining after me, because it will really help to the people who are preparing for this DABT course. So at least they can get some idea about how about the DABT course, and rest of all depends on them, because they have to ultimately put in the efforts to go through all the lectures that we will deliver, and then studying that, and then ultimately passing the DABT exam. Starting with the toxicology definition, you all know that toxicology is the study of adverse effect of chemicals, physical or biological agents on living organisms and the ecosystem, including the prevention and amelioration of such adverse effects. Then toxinology, it is a subdiscipline of toxicology which studies biological exposures to toxins from insect stings, poisonous mushrooms and plants, venomous snakes and aquatic life. Then xenobiotics, you all know xenobiotics is a term referring to substances, whether toxic or not, but they are foreign to a given organism. So as I said, I will be sharing you the notes also with the organizer, so you don't need to note everything down from the presentation. So then coming on to the branches of toxicology. So first is descriptive toxicology. So descriptive toxicology is what a toxicologist in the role of a study director or a study personnel is doing in the lab. So it is concerned directly with the toxicity testing, which provides information for safety evaluation and regulatory requirements. So it includes all the in vivo testing or in vitro testing to support the regulatory requirements. Next, okay, maybe I take a pointer. So next, coming to the regulatory toxicology, it is focuses on ways in which humans and the environment can be protected from toxic effects through regulations and a standard setting. So whatever data the descriptive toxicologists or the toxicologists who do the studies, like the study writer, they provide, the regulatory toxicologists use that information to derive the safe dose levels, and ultimately the aim is that humans are not exposed to the toxic doses over a lifetime. Then moving on to forensic toxicology. Forensic toxicology is concerned with the cause of death from toxic agents, often in instances of drug abuse or misuse. It is a hybrid of analytical chemistry and fundamental toxicological principles. Toxicogenomics is concerned with the compilation and synthesis of information regarding gene and protein expression to understand molecular mechanism involved in toxicology, in toxicity. So far it was more of use for for mechanism of action and other purposes, but now it has been accepted by regulatory agencies also. So recently, EPA has um included toxicogenomics in a 5 day in vivo toxicity assessment, where the animals have to be dosed for 5 days, and then and take out the organs and understand the gene expression. So initially it will be more of an investigative talks, but in the near future, I would see that this will be included in other studies also, to understand the molecular mechanism involved in the toxicity, because right now we are not going into the molecular side. Sometimes it is done when we see some toxicity, and regulatory agencies ask to do some mechanistic tox studies, but in the near future, I would say I see that it will be integrated in many more studies. This is also available. Maybe I will share this EPA link in the notes also, where they have included toxicogenomics in the 5-day tox study. Then coming on to the clinical toxicology, it is concerned with the disease caused by or uniquely associated with the toxic substances. Usually, they are physicians who receive specialized training in emergency medicine and poison management. Then environmental toxicology, it is concerned with the study of the harmful effects of various chemicals, biological and physical agents on living organisms. Ecotoxicology is a subdiscipline concerned with studying the harmful effects of toxicants at the population and ecosystem levels. Mechanistic toxicology is study of how chemical or physical agents interact with the living organism to cause toxicity. It focuses on identifying the molecular and cellular mechanisms by which toxicants exert their harmful effects, including cellular, biochemical, and molecular processes. Then moving on to industrial toxicology. This is a specific area of environmental toxicology that deals with the work environment and constitutes a significant part of industrial hygiene. Occupational toxicology deals with the study of chemicals and other agents in the workplace, worker exposure, safety, and health. So right now, a lot of risk assessors they are deriving occupational exposures. So this is more related to occupational toxicology. Then moving on to computational toxicology, it deals with the use of computational approaches and information technologies to elucidate toxicity. And in the last few years, you all know that the regulatory agencies have started accepting computational tox data in various fields, like for pharmaceutical impurities, ICH-M7 accepts the genotoxicity assessment for using computational tools. For medical devices, the extractable and leachable assessment is done by computational toxicology tools. For agrochemicals, for and especially for metabolites, the European Union accepts computational toxicology tools to do the risk assessment for the agrochemical metabolites, and so on. Then moving on to some of the historical facts. This is just mentioned as text in the Cassette and All book, but I have tried to include some of the pictures to make it more interesting. Also, I am mentioning the numbers here as just to tell you the history. I don't think that you need to remember that when in which year it all happened. You should just know, okay, who was the person and who what was it related to. So, as per the historical facts, I would say this is Shen Nong. This is the one of the most oldest person who was called the legendary king of Chinese herbal medicine and agriculture, and also known as the farmer god. The reason was that he basically taught people how to farm and use the farming tools. He was also said to have tasted hundreds of herbs daily to differentiate the poisons from the medicinal or just plain edible. So I was just going through Google, and he was referred to as God because he had a bull-like face and very huge body. I don't know how much it is true. He's also considered the author of perhaps the world first pharmacological compendium, which is called Divine Farmer's Classic of Materia Medica. Then moving on to the after that, in the ancient Egypt, about 1550 BC. So the Brooklyn Papyrus book, it contained information on snakes and snake bites. So this is just to tell you that, okay, at that time also people knew a lot about toxicology, and it was all documented also. Then in ancient in a- can you please mute, request all the participants. Then in ancient India, Sushruta, around 600 BC, he was an Indian surgeon. His medical and surgical compendium, which is called Sushruta Samhita, foundational work in Ayurveda, contains several chapters related to poisons and poisoning, including descriptions of vegetable and mineral poisons and animal poisons, as well as the advice on medical treatment of snake bites and insect bites. Sushruta Samhita is divided into 186 chapters and contains descriptions of 1120 illnesses, 700 medicinal plants, 64 preparations from mineral sources, and 57 preparations based on animal sources. So you can see this was a quite a descriptive book with a lot of information. Then moving on to ancient Greece. So King Mithridates, sixth from 135 to 63 BC of Pontus. So this Pontus region is right now in the modern day northeastern Turkey. He was called the Poison King because of his family history. So he always knew that he suspected that he would be poisoned by his family members because he was the next higher to the throne. So he regularly ingested mixture of ingredients to protect himself against the poisoning, and attempts to kill himself. Yeah. But as you know, in all this lineages, he was ultimately he ruled for many years, and ultimately people tried to dethrone him. And at that time, you can see in this picture that he had some poison, and when the soldiers came to attack him, these are his daughters. So his daughters told that, okay, you give us the poison first and then you take the poison so that we can die first. So his daughters took the poison and they died. But when he took the poison, the poison didn't affect him because he was already tolerant to the poison. And then soldiers killed him. So, and then because of this name, this Mithridates term was used. This is the practice of protecting oneself against the poison by gradually self-administrating non-lethal amounts. So this is also mentioned in the book. Then you all know Paracelsus. Around the area, around the time from 14 to 1500 AD, he pioneered the use of chemicals and minerals in medicine. And his quote is, you all know, all substances are poison. Only the dose separates a poison from a remedy. So what we call as dose dependence today. This is a term mentioned in the book also, bezoar. Bezoar is a stomach stone, and because those times the poisonings or poisons were very frequently used for killing, so these stones were used as antidotes. So they are indigestible mass found in the GI system, especially in the stomach. So people used to take that, and it used to absorb the poison. These doses were described in ancient medical literature and used as antidotes by physicians. Now, the Middle Ages, Renaissance, that period passed. So around 18th, 19th century, the science started developing, and that was the time of World War. So that time, around 10,000 organic compounds were synthesized by 1880, which included many chemicals like chloroform, carbon tetrachloride, diethyl ether, carbonic acid, etc. Gases like phosgen and mustard were synthesized by 1825 and used as war gases in World War One. Then Orfila, he was a Spanish physician. He used autopsy material and chemical analysis as legal proof of poisoning, and he is called the father of forensic toxicology. Then moving on further, in the 1900s, there was exponential growth in toxicology during World War II area. due to marked increase in production of drugs, pesticides, munitions, synthetic fibers, industrial chemicals, etc. So this is quite interesting. You can go through the links also, which I have mentioned in the notes, if you have time to read this. Yeah. So later on, most of these cases are from US because the book is from US. And yeah, during 18th and 19th century, what was happening is that the US population was moving from villages or small towns to bigger towns. And at that time, uh, there were a lot of quacks who used to practice medicine, who were not properly educated, and also there was a lot of misuse of chemicals and fertilizers. So I'm just presenting few cases. One is this Mrs. Winslow's soothing syrup. So this was used for teething and colic problem in babies. When they develop teeth, so they have pain, and also colic. So this did not need to disclose that its product contain ingredients such as morphine and alcohol. So this drug for children, it contained morphine and alcohol, and 1.5 million bottles were sold annually. Many babies went to sleep after taking the medicine. So normally, when babies cry, we think that okay, they should sleep by any trick. So people who have babies, they understand that. And so when these babies were taking this medicine, they never woke up again, leading to the syrup's nickname, the baby killer. And because many caregivers did not link the death to the syrup, or may have not revealed using the syrup, there is no official statistic of the number of children that died. Estimates, however, put the number at thousands of children who died from overdose or from addiction and withdrawal. So this is all available in Google also if you want to go through that. Then there was another book written by named as The Jungle by an author Upton Sinclair. So this is one of this detail the abysmal working conditions in a Chicago meat packing plant where rotten meat and poison rats were knowingly ground up, canned, and sold to consumers. So this book just created a lot of noise at that time because this showed the really bad conditions which were there in the meat packing plants at that time. So I'm just telling you all these things so that you understand the role of a toxicologist in the society, and how a toxicologist can change the things, and they have changed the things also. So you have a lot of power if you do the right work and you make the right decisions. Then there were other cases around 18 19th centuries also. So lash lure, this was an eyelash product, and in which a number of women suffered injuries to their eyes, including one confirmed case of permanent blindness because of use of an aniline dye, paraphenylene diamine, which is called PPD. And PPD allergy symptoms include localized erythema, pruritus, swelling, or blistering within 48 to 72 hours of exposure. So this picture I have taken from Google only, and this was a case of blindness. Another one is cocaine toothache drops, so similar to the earlier one, Winslow syrup. So here for the toothache, cocaine was there in this drops, and it was given to kids. This is a picture from 1885. All this is available from Google also. Then, because of all these cases that you saw in the previous slides, the US they put this bill or Pure Food and Drug Act of 1906, which prohibited the sale of misbranded, or adulterated food and drugs in interstate commerce, and laid a foundation for the uh, nation's first consumer protection agency, the Food and Drug Administration. So this is how the FDA was formed. This act required that active ingredients be placed on the labels of our drug packaging, because earlier, if you see that the products were containing things like morphine or cocaine or alcohol, but it was not there on the label, and also that the drugs could not fall below purity levels established by the USP. So this is Mr. Ville working in the FDA lab. So they were taking samples from various places and then checking it in the labs. Then coming on to ginger jake paralysis, again the case of adulteration or misbranding. So in US around 1920 to 1933, alcohol was prohibited, which banned the sale of alcoholic beverages having an alcohol content greater than 1.28%. And this is quite surprising, but the reason was that because of the world war at that time, the act was intended to save grain because of the war. At that time, for procuring alcohol, purchasing alcohol, doctor's prescription was required, and pharma companies, what they did was they started selling medicines with higher content of alcohol. And one such concoction was Jamaica ginger. Here you can see the bottle of Jamaica ginger, elixir which contains 70 to 80% alcohol. So you can understand that, and this Jamaica ginger contained nothing, only ginger along with alcohol. And you need, this is a kind of pharma doctor prescription which was required at that time to buy the medicines or alcohol from the pharmacist. This is a picture showing alcohol being discarded in the drain because it was banned. And this Jamaican ginger contained triorthocrysyl phosphate (TOCP), which was an adulterant used in Jamaica ginger, and this was just to fool the regulators because it avoided the smell of the alcohol. And this, by drinking this Jamaica ginger adulterated with TOCP, the people suffered from paralysis of legs, arms, and wrists, and that was known as ginger jake paralysis. So this can be a question about what is ginger jake paralysis was caused because of which chemical. So TOCP is the answer. Then coming on to, this was all pharmaceuticals. So now coming on to environmental pollutants. So Love Canal, I hope many of you would have heard this thing. So Love Canal, which happened around 1950s till 1990s. So this was one of the worst environmental disaster in American history. So what is Love Canal is, Love Canal was an area about 0.28 kilometer square, which was originally meant to be a dream community near Niagara Falls. The vision belonged to William T. Love. So this was the guy who thought that he would make a very good colony or settlement around Niagara Fall region. So what he did was he made some canal around that time, because at that time when he planned this, the electricity was not available. So sorry, the electricity was from the hydroelectric source. So he digs some canal around the Niagara Fall region so that he will use that for generating electricity from the hydroelectric source. And later on, because of some reasons, this canal was just left, and then people started using it for swimming. But then this guy sold it to some other chemical company who used it for dumping a lot of chemicals. So this is the picture about 22,000 tons of over 200 different chemicals were dumped in this ditch from 1940 to 53. So this was used as a dumping ground for chemicals by a chemical company, and later on this guy sold this land to some other person. Before that, he just filled it all with mud. So when this new guy or the locality who bought this land, they were not aware that there are tons of chemicals below the soil. So they started building houses over that land area, and then people started moving in, and there were all schools, homes, etc. on that ground. And after some time, these chemicals which were below the ground, they started percolating and coming above the ground. So people who were living there, they started seeing chemicals all around them in in the pool, in the water, in the playground, and so yeah. So about 100 homes and schools were built at the landfill dumping site, with more than 200 chemicals, and 11 of them suspected carcinogens present in the environment around them. And then there was a lot of movement, and then government came there and they removed the people from that site, but they were already exposed to all these chemicals. And later on, yeah, I will tell you what happened later. So then this is Valley of Drums. This Valley of Drums, this the name came because there were about 2,000 drums dumped into this area. You can see from this picture, officially known as A.L. Taylor Valley of Drums. This is a 23-acre or 9.3 hectare dumping site. It was in Kentucky, US, and it became a collection point for toxic waste starting somewhere around 1960s. And it caught the attention of the state officials when some of the drums caught fire, and it burned for more than a week. Then EPA analyzed the property in 1975, finding high levels of heavy metals, PCBs, and around 150 chemical substances. Then officially EPA started cleanup at that site in 1983, and which ended in 1990. Though still later problems were reported and investigated. So because of this, all this Valley of Drums or Love Canal, the EPA established Superfund or CIRCLA Act. So what is this CIRCLA Act? CIRCLA is Comprehensive Environmental Response, Compensation and Liability Act of 1980. So what EPA did is, in that act they started to take money from chemical industries and all the others who were responsible or might be responsible for environmental pollution, and then using that fund to clean up these sites. So EPA program designed to pay for investigating and cleaning up sites contaminated with hazardous substances. Of the tens of thousands, so there I just gave you example of two sites. So it was not only two; there were thousands of sites selected for possible action under the Superfund program 1178 as of 2024. So there were about 1178 sites which remain on the national priorities list that makes them eligible for cleanup under the Superfund program. So you just need to know what is Superfund or what is CIRCLA. That's all. The Initially it was about $1.5 million fund, and I think right now it is around $8.5 million fund for cleanup of one site, which is designated by EPA. So fine. So this all was happening. So somewhere around 1937, this elixir sulfanilamide tragedy happened. So many of you might already have read about this. So this was called Taste of Raspberries or Taste of Death, and this is the one of the most consequential mass poisonings of the 20th century. 107 patients died from its therapeutic use. This tragedy occurred shortly after the introduction of sulfanilamide, the first sulfa antimicrobial drug, and where the diethylene glycol was used as a diluent in the formulation of a liquid preparation of sulfanilamide, marketed as pediatric elixir. and manufacturing company tested it only for its appearance and taste. So there was no toxicity testing done. Um, so you can understand that at that time there were no toxicity tests available. So people were just introducing their products into the market without any testing and without any taking any approval also from the regulatory agencies. So this is a letter from a lady whose daughter died, and she wrote to U.S. President Roosevelt and described the death of her daughter. And this is just to tell you uh, how how the society or how the people might be affected if the toxicologists don't do their job properly. So what she wrote is, maybe you can read yourself also. The first time I ever had occasion to call in a doctor, and she was given elixir, uh, or Joan. Joan is her daughter. All that is left to us is the caring for her little grave. Even the memory of her is mixed with sorrow, for we can see her little body tossing to and fro and hear that little voice screaming with pain, and it seems as though it would drive me insane. It is my plea that you will take steps to prevent such sales of drugs that will take little lives and leave such suffering behind, and such a bleak outlook on the future as I have tonight. So that is why, regulatory agencies like FDA, EMA, although they request all the data like pharmacology, efficacy data, or DMPK and tox data, but I think the pharmacology or DMPK data is not done in GLP. Okay, although they review it, but always the tox data is done on a GLP, because they are not concerned about if your drug is not efficacious when it comes into the market, because if it is not efficacious, then there is no loss to the patients. Okay, ultimately the company who is developing the drug or anything else like vaccine or something, if it is not efficacious, then the loser will be the company, because if it is not efficacious, ultimately it will be pulled out from the market. But if it is toxic, then it is a big thing. So that is why all the tox studies are done on a GLP, because there should be no manipulation there. Whatever is reported in the tox studies, the regulators want to assess that. Then, yeah, next thing is FIFRA. So FIFRA is Federal Insecticide, Fungicide, and Rodenticide Act. So during World War II, there was a marked increase in the pesticide market. And as wartime research and development produced many chemicals with newly discovered insecticide properties, and these insecticides were not used to control pests. These were for destroying the territories of the enemies. So I will cover that in the next slide also. So when there were a lot of pesticides available in the market, they were also used in the households. So you can see one of this ad. So people were just using this indiscriminately because they found something new which can control pests like cockroaches, etc., in their house. So synthetic organic insecticide usage increased from 100 million pounds in 1945 to over 300 million pounds by 1950. Within 5 years, it increased to threefolds. So that is why this FIFA Act was passed in 1947 to regulate pesticides. Then, with all these issues with the inadvertent and unregulated use of drugs, the FDA Act was passed in 1938. It was also abbreviated as FFDA, FDC, and FDC. But I think if you know FDA Act, that's enough. FDC Act, that is enough. It was set of laws passed by the U.S. Congress in 1938, giving authority to the US FDA to oversee the safety of food, drugs, medical devices, and cosmetics. And within 2 months of passage of the act, the FDA began to identify drugs such as the sulfas that simply could not be labeled for safe use directly by the patient. They would require a prescription from a physician. So, what happened about the sulfanilamide issue, which I presented in the last slide? So, earlier it was sold as a OTC drug, over the counter drug, but FDA said that yeah, you need a prescription for this medicine. And from the time from 1940s to 1960s, there was a lot of abuse because of amphetamines, barbiturates, and then FDA had to do a lot of efforts to control that. And this picture shows the interdiction or prohibition in some venues required undercover tactics, as indicated here by these two inspectors posing as drivers. So FDA inspectors used to go as undercover agents, and then they used to recover all these unauthorized drugs like amphetamines or barbiturates from the market or from entry into the market from the port of entries like that. So coming back to the environmental health. So Silent Spring was a book written by Rachel Carlson in 1962. And because I told in the earlier slide that during the World War a lot of pesticides were manufactured, and these were developed by some countries like Germany, US. And what they used to do is they used to just spray these pesticides all over their enemy territories. US used this a lot over for the Vietnam War also. So when these were available, so farmers also used to spray it inadvertently over their crops. And this was an example of the unlimited use of DDT. So this book documented the environmental harm caused by the indiscriminate use of pesticides. And because this book and this lady, this is Rachel Carlson, so she used to collect the environmental samples, and she found that when DDT was sprayed over the crops or over particular areas, the birds or animals, they suddenly disappeared from those regions. And the birds had problems, like they were not able to reproduce. Their eggs were very fragile, so the babies didn't develop from those eggs. So the environment was completely destroyed where the DDT was sprayed. And because of this lady and because of this book led to a reversal in the US pesticide policy. Okay, I have about 45 minutes left. Oh, sorry, 15 minutes left. Reversal in the US pesticide policy, a nationwide ban on DDT for agriculture use, and an environmental movement that led to the creation of the US EPA in 1970. So you can understand that what an educated environmental activist or a scientist can do. So this lady, her efforts brought US FDA in place in 1970. Then use of DDT was banned in 1972, and it was the first ban of a chemical based on its effect on wildlife. So this is the use of the people who do environmental toxicology. So yeah, so if you are doing the right studies, submitting the right reports to the clients and ultimately to the regulatory agencies, it will ensure that human or animals or environment is are not harmed. And so again, in as I said earlier, because of the heightened public concerns about deteriorating city air, natural areas littered with debris, urban water supplies, President Richard Nixon formed EPA. So here is President Richard Nixon signing EPA documentation. Here is a picture from a plant in Virginia where for decades calcium chloride effluent was dumped into Holston River. Here you see, and in 1970 the company announced, because when the EPA was formed, that it could not meet the new EPA policy, and it would close the plant. So I think everyone knows about thalidomide tragedy in one way or the other. So it was the biggest man-made medical disaster. um, and some people, especially the animal activists, they say that because of after the animal testing also the drugs failed, and they give this as a classical example. But at that time the toxicity testing was not developed, and this drug was tested only for the LD50 test, on the LD50 test, and only the lethal dose was determined. And the lethal dose was around 5000, more than 5000 mg per kg. And they thought that okay, this drug is safe at that time. So there was no repeated toxicity done, no reproductive toxicity done, and this drug was introduced in the market, and none other than by the German pharmaceutical company. Okay, so people should be educated also when they make some claims. And when this drug was introduced in the first in Germany and then in the other European market with only the LD50 test available, between 1957 to 62, there were more than 10,000 babies born with physical abnormalities. All these pictures are available in Google. Not a very good thing to see. And then you all know Frances Oldham Kelsey. She was a young pharmacologist who joined USFDA at that time. So it is all mentioned in Kester Doll also. So she was earlier she was doing some PhD and postdoc in a lab where she was doing animal experiments also. So when the pharmaceutical companies submitted their data to US FDA for approval, so she said no. First of all, I want to review this data properly, because at that time, if you go through that procedure also, so if you submit your FDA application to FDA, you don't need to wait for their approval at that time. Within about, I think, around 60 days or 90 days after submission of data to FDA, you can just introduce your drug into the market. But she said no. So she waited, reviewed all the data which came from Europe, and then she stopped entry of this drug into the US market. And because of this she got the presidential award from John F. Kennedy. So this is another example that how educated a lady pharmacologist or toxicologist or a regulatory agency can help to bring the right drugs into the market so that humans are not harmed. So many people know that thalidomide was banned, but yeah, what was the reason? So definitely there was no testing done. So, and also at that time, the mechanistic toxicity was not developed. So this was the reason of thalidomide toxicity. So thalidomide affects a gene which is called SAL4, SCLL4, and because of this interaction, this gene causes sorry, if there is no thalidomide, then it is normal development, and if the thalidomide is available, then this gene is inhibited, and because of this there are birth malformations. So remember this gene name. So yeah, and if you most of you might be knowing this, but if you don't know that thalidomide is now again approved by US FDA in 1998, and by two companies for the treatment of leprosy, because you know that leprosy is a very difficult disease to treat, but thalidomide is now used to treat leprosy and also cancer, which is multiple myeloma. So it was approved in 1998 by two companies, I think Celgene or BMS, and and you all know as a toxicologist that no drug is safe. Okay, all drugs are toxic. It is only the safety margin which determines the use of that drug for particular indication. So here is a picture, a screenshot from the label of the drug, thalidomide, and it warns that okay, it is a severe life threatening, can cause human birth defects. It is not a OTC drug, Thalidomide is approved for marketing only under a special restricted distribution program approved by FDA. And under this distribution program, only prescribers and pharmacists registered with the program are allowed to prescribe and dispense the drug. So, a drug which was so toxic is now approved by FDA for treating difficult diseases for which was no drug available in the market. So people, I understand that everyone knows that how to go and search for information on FDA website. So you can just Google drugs at FDA, then you will get this window. So if you type the drug name of the drug for which you want some information, then you can just click on search. So here is the name of the drug. So you can see here thalidomide is approved from BMS as well as Celgene. And if you click on one of this, sorry, yeah, so you can go down here. I'm just showing it to the people who might be working with chemical in the chemical domain. So if they don't know about the FDA, how to search there. So here is the label which I showed you. If you go to the review section, here you will get the pharmacology review, and if you go to the pharmacology review, you will get all the tox data also. If you go there, all the reproductive toxicities are done and reviewed by the FDA, and then only here you see the genotox data. And if you go down, you can see all the tox studies, 104 Paraformic CAR-C study, and etc., etc. So you can go through this all this document. So all the tox studies are done and reviewed by FDA, and only after this the drug is approved. Moving back to the presentation. So it's yeah, so FDA Modernization Act came in 3.0 came in December 2024, and as per this bill, it authorizes the use of certain alternatives to animal testing, including cell-based assays and computer models, to obtain an exemption from FDA to investigate the safety and effectiveness of a drug. But it is not for all in all the cases. So FDA will review their data, and it's right now like for small molecules, many of the large molecules also like ADCs, etc., you still need the animal testing. And but this is a recent great news which came out in April 2025, and FDA announces plan to phase out animal testing requirement for monoclonal antibodies and other drugs. So for MABs, they are saying that you don't need animal testing. You can Google it also on yourself. So they are saying the use of with the use of organoids and computational data, they will accept that data for the approval of MABs. So this is available in public domain also. Then some of the additional information or tips, definitions that are given in the chapter. One is hormesis. So exposome, I might take another 5-10 minutes. Exposomes consider the many complex exposures we are subjected to throughout our lives, including diet, lifestyle, and social influences. So the health status of an individual is imprinted by the specific external lifestyle and general external environment together with the internal environment they influence. Big data refers to data sets that are extremely large and require advanced computation to reveal patterns and trends. Open data refers to all the data that can be shared freely by all. So whatever you search on public domain websites like NIH, NLM, etc., it's all open data. Then data science is even more generic term encompassing big data, open data, and more. Then Tox 21, toxicology in the 21st century. There are many publications on this also, and I have shared some references if you want to read further. Is a federal collaboration among the EPA and various NIH branches, aiming to develop better toxicity assessment methods to quickly and efficiently test whether certain compounds have the potential to disrupt processes in the human body that may lead to negative effects. So they are trying to establish more of in vitro or in silico testing to replace animal testing. Then I mentioned this term here, NMDR. So normally most of the toxicologists they know this sigmoid response curve. So I have just tried to explain what is NMDR. So this is a new thing. Which many might be knowing. So NMDR occurs in cells, tissues, animals, and human population in response to nutrients, vitamins, pharmacological compounds, hormones, and endocrine disrupting chemicals. So normally, you know that when we this graph we have seen that we test this MTD dose or a lowest observed adverse effect level or a NOEL, and with these doses we determine the reference dose in terms of in case of chemicals, or maybe AD, PD in terms of pharmaceuticals, etc., or ADI in case of food additives. The terms may be different, but the concept is the same, that we use the tox data to determine a safe dose. And typically this human exposures level should be well below the safe dose. So under the linear dose response model, which we normally do use the high dose testing, because in animal testing we tested high doses, is used to extrapolate to lower doses that are anticipated to be safe for human exposures, which is described here. However, the NMDRCs or non-monotonic dose response curve occur below the toxicological no observed adverse effect level, especially in some cases like this, would falsify a fundamental assumption that high dose hazards can be used to predict low dose safety. So there is a publication. I have shared this link in the notes also if you want to go into the details. So there can be various NMDRCs. So here is one example where the NMDRC is observed above the NOEL level. So this is fine because anyway you are going to use the NOEL for determining the safe dose. So here the NMDRC is occurring below the NOEL. So that is again a problem because you are determining the NOEL to estimate the RFD, and here the NMDRC is happening still below the safe dose level also. So this is again a cause of concern. So there are various examples mentioned in this publication also, which you can go through.